All right, ladies and gentlemen, welcome back to the next installment of Mr. Demont's Lecture Circuit. This is Unit 8, Proteins and Protein Synthesis. Um, can be complicated, but it sort of flows if you get the main gist of how this is working. If you get the backbone of it, then you kind of start to understand the whole thing. So let's dive in. The first part of this actually it's just kind of an overview of what proteins actually do. Like, why are they important? Well, if you look at the A nodes here, um, it's one of the two most important things in your body. Two main components, water and proteins. Why? Because proteins are the ones doing all the work in your cells. They're the ones um, really doing the hard labor here. And what are those major jobs? Look, it's basically everything, right? Structure, transport, movement, defense, which we're talking your immune system response, regulation of literally sleep, energy. Like those are the two main things, but regulation of your bodily function, basically. Um, there's some examples over here. There's some body part examples. Again, it's like it's like every body part, right? It's a ton. It's basically all your organs. They do work everywhere. There's so many different types of proteins. They're just such a major category. Like just saying, here's a protein, you'd have to narrow it down so much further than that. But nonetheless, they're doing a ton. Proteins are all uh, guided by their shape. The shape determines their function. That's kind of their tagline. Their shape, protein shape determines function. Um, the majority of this unit has to do with how do proteins actually get made? Like where does that code lie? And their code for how to make and what proteins to make lies in the DNA. Um, and again, we get into what that process is here in a second, but just a little review about how about proteins here is that remember proteins only function when they're in their uh, conditions that they need in terms of the right temperature, the right pH, not too acidic, not too basic of an environment. That's when they will denature and or lose their shape and therefore are not usable anymore. So something to keep in mind with them. Again, the rest of this has to do with, or the next chunk of this has to do with protein synthesis, which is how are proteins made. Synthesis means to make or to create, right? And so starting off, this molecule that contains the code, that's DNA. DNA has the code to make proteins. Where is it? It's always in the nucleus. DNA never leaves the nucleus. Um, the other organelle that does the work here is the ribosome. The ribosome is the one that actually reads the code, and we'll get into why or how it does that. But it's located in the cytoplasm. That's sometimes you'll hear that translation occurs in the cytoplasm, which is true. Um, but it's the ribosome that's doing the work. Just happens to be well, there's many ribosomes that happen to be doing the work in the cytoplasm. And the DNA is very, very well protected. It has four layers, including nuclear membrane, the nucleolus, that protect it from the outside world there. And here you have the kind of the major steps. I'm going to go through a couple diagrams here. I'm going to draw some stuff to get you an idea of how protein synthesis works here. But this setup right here is the like major steps here. You start with DNA. DNA goes through transcription, which is basically DNA creating an mRNA strand. mRNA then leaves to find the ribosome, where it goes through translation, which is basically a system of an assembly line where they match up with amino acids, and those amino acids then create a string of a protein, right? What we call a polypeptide chain. Um, just to put some diagram to that. So you start, if I was going to have a DNA strand, it would probably look something like this. Usually that's what these diagrams look like, right? So there's your DNA. DNA first, before anything can happen, right? We know that DNA makes copies of itself. For DNA replication, it does that when a cell is ready to divide. In this case, it's not looking to divide, it's looking to um, split so that it can create a copy of mRNA. Now, what has to happen first is it needs to straighten itself out, right? And once it straightens itself out, it can then, I made it a different color. Don't think I'm showing you something different here. But it straightens itself out and it has to unzip basically here. Kind of like, it's basically just like a zipper. It has to unzip down the middle. Once it does that, right, so these are still your DNA pieces here. Then one of those pieces can be used to find those complementary bases. They basically just show up, like those RNA pieces exist, and so they show up as those nucleotides so that you can make those complementary pieces all match up, and you've got an mRNA that has formed here. And when we talk about how they know what pieces to match up, we've talked about this plenty, but let's say, for example, the nucleotides for DNA, we should know these letters by now. G, G, C, A, whatever, right? A, T, C, G, those are the letters. The RNA pieces that match up are A and U match up and C and G match up, right? So I know that this RNA sequence is going to look like this. This U is going to be here. T is going to have an A is going to have a U, C, C, G, U. So you should be able to match up those pieces so that you know what the sequence is on mRNA, right? Um, we'll talk about DNA RNA differences. We've talked about this in class. This shouldn't be news to you about how to match up the mRNA with DNA when that time comes. Um, so you create the mRNA. Again, this is all being done in the nucleus here because DNA cannot leave the nucleus, right? So once this mRNA gets created, we'll bring it over to this next page here. And the mRNA finds its way to a ribosome in the cytoplasm here, right? We'll call this a ribosome. I know it's terrific drawing here. We'll call that a ribosome. So it finds its way here where it is going to meet up with some other pieces here. It's going to flow through basically like an assembly line where it's going to, I'm just going to make up letters here. It's going to flow through where it's going to meet up with tRNAs. Remember those tRNAs, they kind of look like the letter T. Um, they're kind of going through this assembly line as well, where they have the complementary bases, nucleotides to this mRNA. So if they're going in this, I know this shouldn't be upways, it should be sideways, but if UAG is the first three going in, then that's going to match up perfectly with this. Remember, so if this is your first three, this would be your first three right here that are matching up. And then your second three would be, CGG here, or GGC. And so that would be this one matching up with that one. So again, let's label this a little bit here. This is a tRNA. It stands for transfer RNA. This right here, so remember we have a couple other terms here. This is called a codon when it's on the mRNA, and it's called an anticodon. Not that it's like evil or anything, or it's against codons, but it kind of is against them in the sense that it's on the other side of them, but it's the reverse of it, the anticodon, right? So the anticodon matches up with the codon, and then what's really happening here, and the more important piece of what's happening here, is that these tRNAs are carrying with them an amino acid. They each have an amino acid that applies specifically to that codon or that anticodon, right? And so let me go to another page just to get this out of the way. So as those amino acids start to chain together, right, when you get more and more mRNA code being read by the ribosomes, they're going to start to build up and build up until you hit one of those stop codons, and then that's when it knows that the protein is ready. It's made, right? Once that stop codon hits, boom, slices it off. You've got eventually a full scale protein. Sorry, the screen is shaking a lot. I'm trying not to. You got a full-on protein that is ready to go wherever it needs to go, whatever job it needs to accomplish, right? So what are these steps called, real quick? Remember, DNA making mRNA is transcription. It's called that because you're transcribing the code here. You're kind of changing the language here. This step here is called translation. It's called that because you are translating that mRNA code into amino acids and eventually into a protein. Okay, so transcription and translation are the two terms here that we use for these steps. Let's go back to this here. So that should allow it, that kind of, again, it's a little deeper dive into this. Some other things to note is that, again, there's 4 different nucleotides on these. Both DNA and RNA have 4 nucleotides. They share 3 of them. There's 20 types of amino acids. So remember, we've gone through those codon charts and the wheel and stuff. There's 20 of them. Again, amino acids are just the building blocks of proteins. This chart is important in terms of telling you what the differences are between DNA and RNA. Again, DNA is a double strand, right? It's that full double ladder. RNA is just a single strand, right? In this C picture here, you can see the single strand versus the double-stranded A. The bases, the one change, T in DNA, U in RNA. The job is different. RNA acts as a messenger. DNA is just the kind of like base blueprint code. And then their location is a little different. DNA can only stay in the nucleus. RNA can go out into the cytoplasm. The kind of the reason, it's not exactly known why these bases change, but one of the theories here is that thymine is actually too large to fit through the nuclear pores, whereas uracil at the U is not, and it can pass through. So that's why the change happens from DNA to RNA. But that's not necessarily proven to a fault here. In DNA, in a human DNA, average is about 3 billion base pairs, which again, that's a ton. It's only 4 letters making all that base pair action up. You have about 20,000 genes, which a gene is a section of DNA that actually codes for something. So remember, a lot of our DNA doesn't actually code for anything. It's a lot of nonsense, is what it gets called, nonsense codons that they're just there for filler almost, but kind of fills out our DNA. We have a ton of proteins, right? 80,000 to 400,000 different types of proteins, and that those proteins differ from organism to organism, living thing to living thing. We have different unique proteins that are made. What else we get here? So again, this is what I talked about, how a lot of pieces are not needed and are therefore what we call spliced, or cut out, and they get called, pieces that we do need or do use are called exons. So exons are parts of our DNA that we do use. Introns are parts of our DNA that we don't use. They are cut. This diagram, you've seen this diagram before. We've talked about it. You should also have seen it in the practice problem set if you are in fact staying on top of those. But the A, here's your double helix being split, and this is all inside the nucleus. That's what this dark line here, that's the nuclear membrane. C is your RNA. These pieces here are the nucleotides, right? B is talking about a nucleotide. These individual ladder rungs, those are nucleotides, right? Outside here, this is translation out here, where here's your ribosome is the dotted line here. That's what D is referring to. Here's your mRNA strand. It's down here. Okay, it went from C to over here. Here's your tRNA, right? E represents that. F is talking about this. Here's your anticodons here. F is an example of just one of them. And G is your amino acid. Here's the chain being built. And again, that chain will eventually form a protein. Again, codon is a set of 3. So if I had give you a question where I said, um, how many codons to make uh, 3 amino acids, you should tell me it's 3 codons to make 3 amino acids. If I told you how many nucleotides, or if I asked you how many nucleotides would make 3 amino acids, you would then have to multiply that by 3 because it's 3 nucleotides that make up 1 codon. So try to make sure you keep those terms in order here. This, you probably notice that there's this blank section here of um, on the notes here, and that's is for an example for you can do to do this example. I'll do that here. I just got to remember that code and not forget it here. Which, let's see if I can do that. Hopefully I don't screw this up. We'll see. TGG. Oh, I already forgot. I'm second guessing myself. ATC. Okay. So here's your sample DNA strand. And first thing it wants us to do is it says process. So what process is going to turn it into mRNA? That process is transcription. Transcription puts it into what our mRNA is going to be here. And again, that's just base pairing it, matching the base pair. So T is A goes to U, C pairs with G, and you can pair this pretty easily. ACC. A's go to U's, G. I'm moving my face here so I can finish this. UUG, UAG. That would be your base pairs. Hopefully you can understand why these have paired the way that they have. I just took this DNA, transcribed it down here. What do we have next? Codons. So now how do we make that into codons? Well, it's as simple as just putting into groups of 3. So here's your first one, here's your second one, here's your third one, and here's your fourth one, right? Every 3 nucleotides makes a codon, and then I'm not going to go through all these, but it's the process then becomes translation, right? And translation is turning these codons into amino acids and figure out what amino acids they form. And so if we look at the chart over here, the codon chart, I'm just going to do the first one. You can figure out the rest. AUG, again, start the first letter over here, A. Here's the second letter, U, and then the third letter, G. It's where those 3 meet up. First letter, second letter, third letters over here. So A, U, and G. That's MET or methionine would be for the first one. The rest of these you can probably figure out on your own if you have not already. And then the last part of B here, and C and D is going to fly by. I've been talking for a while, but is these 2 types of mutations. Now, we talked about mutations before a little bit, but let's make sure you understand the difference between a point mutation and a frame shift mutation, which a point mutation, I'm just making up letters here. A point mutation is when you have changed one letter. You just changed it, right? And you can see if I go to the notes instead, it says a nucleotide is changed. The gene usually stays the same and it rarely causes problems. That could be as simple as ACTCCG changing to um, ACACCG. Obviously, one letter has changed. It can make a difference. It can change a protein or a gene, but it's much more unlikely to because it's just one letter change. It hasn't changed anything after or before. So, whereas this one section would be affected, there's many, many more letters after that are still all the same and unaffected. So it doesn't have as large of an effect. Again, this is a point mutation. It's a point mutation because it's saying at one point on the DNA it's changed. A frame shift mutation is a little different. Frame shift is much more harmful potentially. A frame shift mutation. Sorry, I'm shaking the screen again. is one where a nucleotide is added or deleted. The gene is always going to change because if you think about it, right, if you have, I don't know if I'm doing the same one. I think it's the same one I had. I'll add a few more just to kind of show you what I'm talking about here. So if this is my sequence, and let's say I remove this C right here. So now I have A, T, C, C, G, A, C, C. Now, it doesn't look like it's changed all that much. The problem, though, is if you remember how DNA gets read, DNA gets read in sections of 3. That doesn't change just because of a mutation, right? Sections of 3. Now your sections of 3 are ATC, CGA, CT, whatever comes next, and say it's like G or something. Now every single one of these codons have changed, and every single one is going to change from here on out because you've taken a letter out. It's like reading a sentence where, like, the cow ate the bug, right? If you switch one letter, like change this E to an A, you could probably still read that sentence. Try that sentence again if you completely shift everything over now. Let's say you take out that E completely, but you still have to read it in segments of 3. It's a little more difficult, right? And now if you're looking for that, you know that's what happened, it's a lot easier, but obviously, if you have to read it in segments of 3 like DNA gets read, it's, you know, becomes a little more dicey. So that's why frame shift is so much more, dangerous here. Um, but anyway, let's roll through here. That's A and B. C and D is gonna be really quick here because we're just talking about now, and this with technology, DNA technology. And it's used for different things, these different types of technology, but the majority of it, we're talking about how to extract DNA or how to study it, um, which we've talked during class briefly. I showed you a video about the Human Genome Project, which was a huge undertaking in the 90s. It was kind of like sort of comparable to Race to the Moon, like US and Russia, but this was by two companies that were studying, or two research centers that were trying to figure out the full map of human DNA, um, to for these possible uses here. You can see why it would be so important: predict illness, cure illnesses, identify disease genes, genes that causes whatever. It was really important work. It basically changed the face of genetic illness and medicine, and was a huge boost. Gave us a lot of things that we did not have before. And listed here, it's a bunch of technologies, right? Some a little bit we talked in class, not a lot, but the main thing, and something that kind of ties a lot of these together, is this first thing that says restriction enzymes, which a restriction enzyme is just an enzyme that is used in some of these to basically cut particular pieces of DNA that you need. It can be either useful pieces or just cutting the pieces that we don't need, like the nonsense pieces. So it'll cut specific pieces, right, that you need, that you want to study or that you want to measure. Um, Electrophoresis, we talked about in class a bit back, how it's this gel, and you put the pieces, these fragments of DNA into or these pieces of DNA into these wells, and it uses electric charged electrodes to, basically like a magnet, to send the bigger and smaller pieces to along this chain here, so you can compare them. Um, RFLP, Southern blot, Western blot, those are just different uses for uh, inside of electrophoresis. Different, you can read through them. I don't It's not necessarily important. It's just different variations inside of there and different uses for them, but you can sort of read through. Plasmids. A plasmid is a technique based, basically where you're adding a piece of bacterial DNA to another type of DNA, just any other type of DNA, not bacteria. And it can be used for making DNA really, really fast because bacterial DNA copies very quickly. So plasmids can be used to combine with them to sort of generate really quick turnover, really quick copying of DNA. You can see why it's used. This a lot of times is used in antibiotics to make antibiotics much more quickly. Um, GMOs or genetically modified organisms are used in a lot of different ways. Um, that's a whole discussion in itself. We don't need to get into it. Just understand that these are organisms that are changed. Their DNA is changed specifically for a purpose, whether it's when you're breeding animals or um, plants, crops that you're trying to make bigger, smaller, sweeter, whatever. Um, PCR, again, I told you we're flying through this section. PCR or polymerase chain reaction is you'll have a question on the test about this where uh, it's a technique where you're trying to study or test DNA, and this technique allows you to make a lot of copies of it pretty quickly. Um, Basically, by sort of doing what I talked about before just a minute ago, where it cuts certain pieces of that DNA and like certain pieces that you're trying to actually use, and it puts them in this chamber where it heats it to break it apart. It cools it and allows pieces to kind of copy. And so it's making copies by heating and cooling and heating it again and cooling it again. And so it's making like a million copies inside of these cycles. And therefore, the more DNA means it's just easier to use in the study. So that's kind of what PCR. That's why you see this diagram of like you start with one piece, it's going through, it's creating more pieces as you go through, and then this would actually go on for like 20 more arrows here, just creating more and more copies of this same fragment of DNA. You have this magic bullet gene therapy. Gene therapy is something that they've started using in cancer treatment, where they are using viral DNA to help basically trying to. I'll get into how viruses work here later, but basically try to get that viral DNA to like insert itself into the machinery of your cells and to help do something or make something that that your cells are not doing very well right now or not doing at all. So it's like it's more complicated than what I'm making it seem, but it's basically just using viral DNA to try to jumpstart your certain processes in your body. CRISPR is another one or the last one here, where um, kind of similarly to PCR, it basically it uses it's a system or a setup that cuts out bad DNA sequences in terms of it locates these bad sequences on your DNA. It goes through a couple steps, but it basically cuts it out to try to get rid of it, to get rid of malfunctioning or bad or mutated DNA. Last but not least, here we're talking vaccines. How do vaccines work? Well, your vaccines work depending on what is in that vaccine. Generally, it's a either partial piece of a virus or a weakened version of a virus, where it's a similar type of virus that is injected, and it's injected to generate an immune response. Um, Usually, that immune response can kind of give you some symptoms depending on how your body reacts to it, but um, it could give you a fever, inflammation. That's a good thing. That means that it's actually working, and that means that your body's immune system is responding to it. Now, on the flip side, just because you don't get side effects, that doesn't mean that it's not working. Um, just mean that your body is just a little bit more resistant to side effects. Um, how do they cause disease? Well, how do viruses work? So viruses are um, tricky because they're not technically living things, but they do act in a lot of ways like living things in terms of having their own DNA or RNA, and they basically take over cells and our cells generally don't have the means to fight these viruses off very quickly. So it takes a while for our body to react, or sometimes we can never get rid of or never shut down a virus. But it can cause cells to burst. It can basically causes your cells, your immune system to attack itself. Um, and again, like it says here, they replicate very quickly. So it's difficult to completely get rid of them or to completely destroy them. So down here it talks about how vaccines work. We already talked about that. So how kind of on the topic of COVID-19, COVID-19 is an RNA vaccine, so it's a little bit different here um, in terms of how it works. It's not actually You're not actually injecting the virus into you in terms of how it normally happens. It's actually just the RNA um, of, again, it says of one viral protein, so it's a part of it, but it's just the RNA. So since it's the RNA, it's not actually able to completely replicate at all, and it doesn't. So there's zero chance that it could ever lead to COVID-19. Yeah, it could give you some symptoms, and it's not very pleasant, as I can attest to, but could never possibly turn into the actual virus. Um, so you can see the body does not accidentally make more viral DNA. That's not possible. Um, Body responds to the portion of the virus it would see in a real infection. So it gives you that protection. Is it 100% effective? No, it's not. Um, Very few vaccines are 100% effective, but the reason that some of these viruses have completely been eradicated is because of this theory of, or not theory, but this principle of herd immunity, where if we've um, vaccinated so many people that the statistics or the percentage of people having it have gone so low and have so much protection that it'll eventually just wipe itself out. And that's why, if you're unsure, if as you see information about, should I get the vaccine, should I not get the vaccine, you should. And it's because whether you think it works or not, it's this herd immunity thought, that's the principle of that will eventually help us get rid of COVID-19. And that does it. Let's end it there. The MDLC version, volume 8. That's a wrap.